my dear friends and professor. Good evening. My name is Jasprabjot Kaur and I'm going to give an oral presentation on the article named Fermentative Hydrogen Production from Microalgal Biomass by Single Strain of Enterobacter aerogens, Effect of Operational Condition and Fermentation Kinetics. Let's see the outline. I'm going to discuss about the introduction, materials and methods, methodology, results, discussion, and future direction in my upcoming slides. First of all, the introduction. I'm going to start with the introduction by revealing that the biohydrogen production through dark fermentation is a promising technology for generating renewable energy while using microalgal biomass as a third generation feedstock can further increase the sustainability of the process. So, 5 hydrogen-based technologies have been identified in Europe. The European Strategic Energy Technology Plan has identified hydrogen-based technologies among the technologies needed in Europe. Hydrogen reduced local air and decarbonization in Europe transport system. The question comes to our mind that why hydrogen is the only which can be used to reduce local air. So here we have answer. Hydrogen can play an important role in the reduction of local air pollutants as well as decarbonization of the Europe's transport system. At present, it is produced from the fossil fuels, coal gasification and the natural gas steam reformation. It is also produced from the water with the process of electrolysis and photolysis. Moreover, it is more efficient and environmentally friendly process of production of hydrogen. Now let's have a look at the hydrogen production through the anaerobic fermentation. Why we have used this topic? Because the production of hydrogen through anaerobic fermentation is a relatively simple method, which is useful in providing species wide spectrum of substrates including waste products. And the microalgae such as, let's have a look at the next slide so that we can have an exemplification. Scindesmus obliquus, Nanochloro species, Cholera vulgaris, Dunaliella tertio-leca, and Spirogyra species. Back to the slide. Enterobacter aerogen is the best biohydrogen production bacteria. Why? Because it is most widely used in studying model strains. It can grow under oxygen and hence making manipulation. Manipulation means changes in the bioprocess easily. Why it is more effective bioconversion of microalgae? Because these bacterium as known as the widely studied strain, which are able to grow in the presence of oxygen and therefore makes the manipulation process much easier. Hydrogen production through dark fermentation can be explained as it's a complex system where environmental and the operational factors such as temperature, percentage of hydrogen, and partial pressure of hydrogen can be regulated. Why? This dealing complex substrates as a low partial pressure which facilitates the mass transfer of hydrogen from gas phase. The biohydrogen production is also influenced by inoculum sources, enrichment, inoculum procedure, and the bacterial culture seed. Why? The main aim of this work was to explore the operational conditions which were the best suited for the biohydrogen production using the Scindesmus obliquus species. And this flowchart is revealing the biohydrogen production process in a much easier manner. So here we can see that it is split into 3 components: fermentation, biophotolysis, bioelectrochemical system. Next, operational condition for the biohydrogen production. For this, we need various materials and methods, which includes the initial gas-liquid volume ratio, bacterial growth and inoculation procedure, sterilization, fermentative kinetic parameters. Under the initial gas liquid fermentation, the serum bottles were filled with a determined volume of liquid media. And in bacterial growth and inoculation procedure, there is a facultative anaerobic bacterium named Enterobacter aerogenes. In sterilization, the sterilized liquid phase was centrifuged and the supernatant collected for analysis. And for the fermentation kinetic parameters, these were carried in volume by volume ratio, that is gas liquid ratio of 5 and the bacterial inoculation of 10%. Now let's come to the result and discussion. The gas liquid volume ratio is a relatively operational parameter to be taken into account in dark fermentation hydrogen process. Why? Because it is more effective in dark fermentative hydrogen process, which helps in the increase in the gas liquid ratio and increases biohydrogen production. And the question comes to our mind that what's the effect of bacteria growth and inoculation condition? Enterobacter aerogen grows well under aerobic condition while using with the help of using peptone water, and hydrogen can be obtained in a higher manner. Higher bacterium mass was obtained through aerobic condition. And we can well explain this with the help of the bar graph representation, which is showing us the various gas liquid ratio and the hydrogen yield in milliliters per gram of alga. And the effect of sterilization is a microalgal in a complex substrate that requires the harsh penetration to yield the high hydrogen production. Why? Because the bacterial growth and inoculation condition have a major impact on the production of biotechnical process of Enterobacter aerogenes. And the biohydrogen production kinetic can be like operating conditions were favorable to the microbial metabolism, and it is more advantageous as it reduces energy expenditure. And this linear graphical representation is providing a clear-cut explanation that how biohydrogen fermentation kinetics of microalgal biomass by nitrogen happens. So what is our conclusion? We come to our conclusion that the best determinants of biohydrogen production is the bacterial growth and inoculation condition, the effect of initial gas liquid volume ratio, sterilization procedures, and the fermentative kinetics, which helps in yielding maximum hydrogen production by using the biomass. If we come to the future direction, we can conclude that these results can be useful for providing insight and extrapolation to dark fermentation and microalgal species. Thank you. If you have more questions, you can ask. Thank you.